Good evening, and welcome to tonight's special segment on the incredible processes that sustain life on our planet. Tonight, we turn our focus to the Calvin cycle, a fundamental part of photosynthesis that converts carbon dioxide into organic compounds. Understanding this process is vital not only for grasping how plants grow, but also recognizing how they contribute to our ecosystem and help mitigate climate change by absorbing carbon dioxide.
The Calvin cycle, often called the light of independent reactions, occurs in the cytoplasm of plants. This vital process allows the plants to transform in sunlight into energy, ultimately supporting both the plant growth and the broader ecosystem. Importantly, it utilizes ATP and NADPH, which are produced during light-dependent reactions of photosynthesis. A Calvin cycle consists of three main stages: carbon fixation, reduction, and regeneration. The cycle starts when the carbon dioxide enters the leaf through tiny openings called stomata. An enzyme known as rubisco catalyzes the reaction between carbon and dioxide and ribulose bisphosphate, or RUBP. This reaction forms a six-carbon compound that quickly splits into two molecules of 3-phosphoglycerate or 3-PGA.
Let's break down the stages of the Calvin cycle, starting with carbon fixation. In this phase, three molecules of CO2 is combined with 3 RUBP, resulting in 6 molecules of 3-PGA. The first phase, carbon dioxide fixation, basically captures the CO2 from the atmosphere, so it can be used in the reactions. To do this, CO2 is attached to RUBP, a 5-carbon molecule. The enzyme used in this reaction is called rubisco, and the result of the reaction is an unstable 6-carbon molecule that quickly splits into two 3-carbon molecules called 3-phosphoglycerate or 3-PG.
I find it fascinating how this initial step is crucial for capturing carbon dioxide which plants need to grow. Without this process, plants wouldn't be able to produce the food that sustains us. Exactly. Now, the next stage is the reduction phase. Here, it uses ATP and NADPH, which are produced from light-dependent reactions. Convert 3-PGA into glyceraldehyde 3-phosphate or G3P. Notably, one G3P molecule exits the cycle to eventually form glucose, which serves as an energy source for the plant.
Next, the process of converting the CO2 to carbohydrate begins. This phase is called the carbon dioxide reduction phase because we're adding electrons and energy to the CO2 molecule. During this phase, a sequence of reactions uses NADPH and some of the ATP from the light reactions. These molecules supply the needed electrons and energy for the CO2 reduction. Electrons are added from NADPH, and through a series of reactions, 3-PG is reduced to form G3P, a carbohydrate. ADP and NAD+ return to the thylakoids to be converted back ATP and NADPH with the light reactions. One of the G3P molecule is set aside as a building block for glucose, with the majority of the G3P molecules move forward into the third phase of the Calvin cycle.
In this stage, the energy captured from sunlight is utilized to transform 3-PGA into G3P. This is where the magic happens. Energy from the sun is turned into a form that can be used by the plant.
The ability of plants to convert sunlight into chemical energy is remarkable. G3P is a building block for many important biomolecules, including sugars and starches, which is essential for plants' metabolism and growth.
Finally, we have the regeneration phase. The remaining 5 J3P molecules are used to regenerate RUBP, allowing the cycle to continue. This step requires the additional ATP, ensuring that the cycle can keep running efficiently.
In this phase, ATP is used to combine the rest of the G3P molecules to form RUBP molecules. This RUBP can then combine with additional carbon dioxide molecules, continuing the carbon reactions. To form a glucose molecule, the cycle actually has to turn 6 times because each turn of the cycle adds only one carbon atom from the incoming carbon dioxide.
It's important to remember that the Calvin cycle is dependent on the light reactions to provide NADPH and ATP, which in turn provide the electrons and energy needed to form carbohydrates.
Understanding the regeneration phase really highlights how efficient plants are. They continually recycle their components to keep the process going. It's like a well-oiled machine. And it's not just about plants. This cycle is crucial for all life on Earth because it's the foundation of the food chain.
In summary, the Calvin cycle takes in carbon dioxide, ATP, and NADPH to produce G3P, which can be transformed into glucose and other carbohydrates.
Summary of key findings on the Calvin cycle. Definition and importance. The Calvin cycle is a crucial component of photosynthesis, allowing plants to convert carbon dioxide into organic compounds. It plays a vital role in plant growth and helps mitigate climate change by absorbing carbon dioxide. Location. The cycle occurs in the stroma. Main stages. Carbon fixation. Carbon dioxide combines with ribulose bisphosphate to form 3-phosphoglycerate through the enzyme Rubisco. Reduction. ATP and NADPH convert 3-PGA into glyceraldehyde 3-phosphate or G3P. Some G3P exist to cycle to form glucose. Regeneration. Remaining G3P molecules are used to regenerate ribulose bisphosphate, allowing the cycle to continue with additional ATP required. Key molecules. Inputs: carbon dioxide, ATP, and NADPH. Inputs are the substances or energy that are required for a process to occur. Outputs: G3P, which can be converted into glucose, 3-PGA as an intermediate, and regenerated ribulose bisphosphate. Outputs are the substances or energy that result from a process. Ecological significance. This cycle is foundational for energy production in ecosystems and supports the entire food chain.
With insights from experts and students alike, we see how the Calvin cycle is not just a biochemical process, but a vital part of our ecosystem.
To conclude, the Calvin cycle is essential for the growth and survival of plants, forming the basis energy production in most ecosystems. Its 3 stages, carbon fixation, reduction, and regeneration, highlights the intricate interplay of sunlight, carbon dioxide, and plant biochemistry. As we continue to learn about these processes, we gain a deeper appreciation for the natural world and the vital role that plants play in sustaining life on Earth.
Thank you for joining us as we explore the wonders of the Calvin cycle. Stay tuned for more engaging science stories that shed light on the processes that sustain life on our planet.